Assalamu alaikum. So we have this short lecture on cell-cell interaction. فالمرة الماضية حكينا إنه cells can interact with their surroundings على الأشياء اللي حواليها. So they can interact with matrix proteins, but they can also interact with each other. And there are different ways by which they can interact with each other. We're going to talk about some of these mechanisms of cell-cell interactions. So we have basically different types of receptors on the cell surface. في أربع أنواع. So primarily we're going to focus on these four. We have receptors or adhesion proteins known as selectins, and they can interact with carbohydrates on the other cell line or cell type. We have integrins, and we talked about integrins كتير. We have and integrins, there are different types, like they can be members of immunoglobulin superfamily. We have the immunoglobulin superfamily. That can include integrins, but also they can include other proteins. And they can interact with each other with like proteins. يعني بيعمل interaction مع بروتين من نفس الفصيلة نفس النوع. And we have cadherins. Now, integrins, we talked about them before. Oh, and cadherins, برضو, they do homophilic interactions. Now, integrins, we talked about integrins before. They exist in focal adhesions and hemidesmosomes. And we talked about cadherins. They exist in adherens junctions and desmosomes. But let's talk about selectins first. So selectins exist on the surface of leukocytes, immune cells. As they are swimming in blood, they can interact with endothelial cells. Endothelial cells اللي هم lining up the blood vessels. Now, the surface of endothelial cells contain receptors and sugars as well. So these are glycoproteins on the cell surface, and they are glycosylated. Now, the interaction happens between the selectins on the surface of leukocytes with the carbohydrates on the surface of endothelial cells. On these, the carbohydrates associated with the glycoproteins, membrane, transmembrane glycoproteins. Once they do that, stronger interactions take place between integrins on the cell surface of leukocytes with intracellular adhesion molecules. Intercellular adhesion molecules or ICAMs. هدول, they exist also on the surface of endothelial cells. Okay, so initially, it's like leukocytes, they're swimming fast, and then interaction happens between the selectins and the carbohydrates on the surface of endothelial cells. So these leukocytes slow down. When they slow down, you have another interaction that takes place between the integrins on the surface of leukocytes and ICAMs on the surface of endothelial cells. So note the interaction is heterophilic. يعني selectins interact with something else. Once this interaction takes place and leukocytes stop, they can extravasate. Extravasate ايش يعني؟ يعني انه they leave the vasculature. They leave the vessels. They move between endothelial cells into the tissue. Okay, and they do whatever they do. فهدول السلكتنز. Okay. طب cadherins. Cadherins form homophilic interaction with other cadherin molecules between cells. And the interactions between cadherins take place throughout development and throughout our life. So they are physiologically important. So these interactions take place between embryonic cells, and they play important roles in development and differentiation. They can also form between different nerve cells in the synapse. Cadherins, the interaction between cadherins can also maintain the stable junctions between cells and tissues. يعني you have the formation of adherens junctions and desmosomes. We'll talk about them in the following slide. لكن I want you to know that there are three types of cadherins. We have E cadherins, N cadherins, and P cadherins. E cadherins are found in epithelial cells, and that's why they are known as E cadherins. N cadherins exist in neural cells, N cadherins, and P cadherins exist in placental cells. There are other types of cadherins like desmosomal cadherins, 7 transmembrane cadherins. We're not going to talk about them, okay? We're not going to talk about these three. I just want you to know that. Now, why do I want you to know E cadherin? Because it exists in epithelial cells. So it allows cells to connect with each other. If they lose this interaction, they become mesenchymal-like. They become elongated. They become motile. So E-cadherins prevent cells from moving. E-cadherins, when they interact with each other, they stabilize cells to remain in their places. Okay, they do not move anymore, or they do not move. Once they lose this interaction, then they become mesenchymal and they can move. And this is exactly what happens in cancer cells. So once you have this cell-cell interaction, you have a tumor mass, but the cells are interacting with each other. They do not move. They remain in their place, and this is what we call carcinoma in situ. That is, you have a tumor mass, but it stays in place, in situ, in place. But once they lose E-cadherins, they become mesenchymal-like, and then they can migrate and they metastasize. So let's talk about adherens junctions and desmosomes. So just to remind you, in adherens junctions, you have the cadherin-cadherin interaction, homophilic interaction, between two cells. So here you have cell number 1, here you have cell number 2, and here you have this interaction between the two cadherins coming out from each one of these cells. The cytosolic site of the cadherin interacts with actin binding proteins, and these interact with actin itself. Note that the cytosolic side of cadherins interact with catenins. Again, why do I mention these proteins specifically? Because they are biomarkers for cancer development, cancer formation, for progression of cancer. So adherens junctions, you have cadherins, and these cadherins interact with the actin filaments indirectly via catenins and actin binding proteins. Note that, so you have this interaction between actin filaments in two different cells, but they are neighboring cells. نحكي adjacent يعني قراب على بعض, very close to each other. Now, desmosomes is you have desmosomal receptors, or so desmosomal cadherins, and these cadherins برضو you have You can have heterophilic interaction between different desmosomal cadherins. Now, instead of interacting with the actin filaments, you have the interaction with the intermediate filaments. And this is mediated by intermediate filament binding proteins. So again, you have the interaction between two cells via desmosomal cadherins, and the interaction links the intermediate filaments between two adjacent or neighboring cells. I want to show you something real. These are images generated from my laboratory. What we did is that we stained a desmosomal cadherin protein known as desmocollin, and we also stained for actin. Actin is in red, and the desmocollin is in green. The reason why we did actin is just to show cell shape. Okay. Now note, in green you have the desmocollin, this desmosomal cadherin, present between the cell-cell boundaries at the points of cell-cell interaction. And you can see it all over the place. That is in every cell-cell interaction, point of interaction of two cells, two adjacent neighboring cells. When we took these cells and we treated them with the hormone dihydrotestosterone, the cells became mesenchymal-like. They became elongated. They were able to move. They become motile, and again, there are no desmosomes. It's gone, and it's not organized anymore in these elongated cells. Now, note cells that were not affected by the hormone and cells that maintained their interaction with other cells. You still have these organized structure of desmosomes, and you still have these the presence of desmocollin at the cell-cell interaction points. So this is just to show you something really, I mean, something not in your textbook. Okay, so let's move on. So this is something extra, okay? So cells can interact with each other at points, regions known as tight junctions. So in tight junctions, you have this sort of like a belt. You have this strong interaction between proteins that exist on the cell surface of two adjacent neighboring cells, and you have this this interaction that seals the spaces, بيسكر the spaces between the cells. The purpose of tight junctions is really to two things. Okay, what it does is that it separates the spaces outside of the cell. So right here you have these tight junctions, interactions, and molecules, small molecules, ions, for example, cannot pass between cells. So these spaces here are closed. So whatever is in the lumen, for example, cannot pass between cells to the basolateral surface. That's one. Two, tight junctions also close or separate the apical region from the basolateral region. So if you have proteins that exist at the basolateral surface, and since we know, proteins move within the membrane, these proteins cannot pass through. So apical proteins remain in the apical part of the cells, and basolateral proteins also remain at the bottom of the cells. Okay, so they cannot move upward. So that's the function of tight junctions. Now, an example of a tight junction protein is a protein known as claudin. Claudin. Okay, so right here, claudin. And it turns out, there is a type of breast cancer known as Claudin low breast cancer. يعني breast cancer that has low expression of Claudin, the protein Claudin. This type of breast cancer is known to be mesenchymal-like, يعني, meaning it's motile, elongated. It has low ECadherin expression. Patients with Claudin low breast cancer have poor survival. It's an aggressive type of cancer. They have poor prognosis. يعني إنه the outcome of the cancer is bad. They do not survive for a longer time. Um, these cancer cells metastasize. Usually they are metastatic and they occur at younger age of onset, unfortunately. It's very common to see tight junctions, adherens junctions, and desmosomes all together in two cells or between two cells. This is an electron microscopic image. But here you have cell to the right, cell to the left. Here's a tight junction. That's the seal. Here you have adherens junctions right here. And here you have desmosomes right there. So you have this interaction with the intermediate filaments right here. Here with the actin filaments. So you have, again, the tight junction. That's the illustration, tight junction. You have adherens junction, actin filaments. You have the desmosomes, intermediate filaments. Okay, so usually you have these associated together. Another mode of interaction between two neighboring cells is something known as gap junctions. And gap junctions are basically channels that connect two cells to each other. So right here we have one cell, here we have another cell, and you have this channel that connects the two cells together. So something small, small molecule, ions, for example, signal molecules can pass through the channel from one cell, and these molecules can reach the other cell, going out from the other end of these channels, gap junctions. And these gap junctions are known as connexons. Connexons. Now, each connexon is made of 6 transmembrane proteins known as connexins. Okay, connexins. So 6 of these connexins come together, forming a connexons, and these connexons form a channel eventually. Now, there are different types of connexins, by the way, and you can have combinations of these connexins that make up connexons. And you can have connexons forming of different connexins forming this channel, by the way. Now, there are specialized connexons that exist, for example, in the nerve cells, between nerve cells. So what happens with nerve cells is I have a presynaptic nerve cell that releases a neurotransmitter. That neurotransmitter binds to the receptor on the cell surface of the postsynaptic nerve and transmitting a signal. Well, what happens sometimes is that between these nerve cells, you can have these gap junctions where the charges can, the electrical charges can pass through the gap junctions rather than being transmitted via these neurotransmitters. So these are specialized gap junctions. Now, defective connexins or gap junctions are associated with a number of diseases. Examples include Mary-Charcot tooth disease. Whoa, we talked about this disease a lot, actually. So deafness, skin disorders, cataract. Okay, and actually, we have cases of hereditary deafness, summum متوارث. And it exists in certain villages here in Jordan where you have يعني relatively abnormally large number of children being deaf. Um, so it is hereditary, and it turns out that that they carry mutations in the connexins. So, but there's something يعني شوية puzzling, you know. Although connexons are found in many tissues, only a limited number of tissues are affected. And this is puzzling. Well, why is it that you have two tissues, different tissues, they express the same connexin, yet one of them is affected but the other is not? And there are several reasons for why. One, compensation by other connexins. So basically, if in one tissue, a certain tissue would have the ability, if one connexin is downregulated, if it is not expressed, it can compensate by expressing something else, another connexin molecule. Something else is that the affected tissue requires this particular connexin, okay, but the other tissue may not require that connexin. It doesn't play a major role. It doesn't have a strong functional purpose in that tissue. It can be that mutations affect the proper assembly of connexins in the Golgi apparatus. So for in certain tissues, so it depends on the mutation as well. That is, yani mutation in one connexin can prevent the formation of a proper gap junction, a proper connexon within one cell, trapping the connexin or the other connexins in the Golgi apparatus. So these are different reasons for why you can have mutations in one connexin, but you have certain tissues being affected. Again, just to sum up, so we have different ways by which cells can interact with each other and their surroundings. We talked about focal adhesions, where you have integrins linked to the actin filaments, and the integrins bind to the extracellular surface. We have other types of proteins that form hemidesmosomes, and these hemidesmosomes can interact with other integrins that can interact with the filaments, with the intermediate filaments. We have cell-cell interactions that take place via tight junction, sealing the spaces and sealing the membrane. We have adherens junctions, cadherens forming interactions, homophilic interactions between two cell types, and these cadherens interact with the actin filaments. Then we have desmosomes, right? So you have these interaction between different desmosomal cadherens, and they interact with the intermediate filaments. And we have gap junctions. We have these channels as well. Okay, so things can move from one cell to the other. Allah